Hello students, let us now discuss on the topics lipids. Introduction. Lipids cannot be defined based on the chemical nature of the members since they differ widely in structure and possess different chemical groups. But they are commonly soluble in certain nonpolar organic solvents. Based on this, lipids have been defined as compounds soluble in ether, chloroform, and the benzene. Moreover, almost all of the lipids are soluble in petroleum ether. Lipids are greasy substances present in both plants and animals and constitute as main components of foods along with proteins and the carbohydrates. Classification. Different authors adopt different systems for classification of lipids. We are narrating a system of classification consisting of 4 classes such as simple lipids, compound lipids, derived lipids, and lipids complex to other compounds. Structures of different lipids and some related narration. Before we discuss about the structures of higher lipids, we need to know the structures of fatty acids, which are derived lipids. Fatty acids are carboxylic acids which take part in the formation of other lipids such as triglycerides and the phospholipids, etc. Depending on the number of carbon atoms they have, they may be even carbon and or carbon acids. Even carbon acids are generally found in natural fats. However, certain plants and animals fats have odd carbon acids. Short chain, medium chain, and the long chain fatty acids have 2,2,6, I2,14, and 16,2,24 carbon atoms respectively. Very long chain fatty acids have more than 24 carbon atoms. Moreover, based on the nature of hydrocarbon chain, they may be saturated, unsaturated, branched, and the hydroxyl derivatives. Numbering of carbon atoms of fatty acids can be done starting from carboxyl group carbon or from terminal methyl group carbon atom, denoting it as omega 1. Next carbon atom is omega 2, and so on. In the former case, the position of double bond is indicated by prefixing number in the systematic name. In the latter case, if the fatty acid is referred to as omega 3, then it indicates that this fatty acid has a double bond between omega 3 and the omega 4 carbon atoms, as one nearest to omega 1 carbon atom. Linoleic acid and linolenic acids are essential fatty acids because they cannot be synthesized in our body. Arachidonic acid becomes essential when its precursor linoleic acid is not sufficiently obtained from the diets. Next, simple lipids. They are esters of alcohols and fatty acids. If the alcohol is glycerol and it is esterified by 3 fatty acid molecules, they are triglycerides. Fats and oils are triglycerides. On the other hand, esters formed by long chain alcohols comprising 16 to 30 carbon atoms and long chain saturated and unsaturated fatty acids comprising 14 to 36 carbon atoms are wax. Examples of wax are beeswax, lanolin of lamb's wool, and the spermaceti oil of whale, etc. Wax are used in the making of candles, cosmetics, lubricants, ointments, and the polish, etc. It can be noted that fatty acids composed in fats vary widely in chain length. Thus, molecular weights of different fats vary. We can have an idea on this by determining their saponification number. Saponification number is defined as the amount of potassium hydroxide in milligram required to saponify 1 gram of fat. We know that a molecule of fat is saponified by 3 molecules of potassium hydroxide. Since a gram of fat composed by low molecular weight fatty acids has more of its molecules, requires more amount of potassium hydroxide for complete saponification. Thus, saponification number is an index of average molecular size of fatty acids of fat. It can be experimentally found out. Next, iodine number. Halogens can be added to the double bonds of unsaturated fatty acids of fats. Iodine number expresses the amount of iodine in gram added to 100 gram of fat. This can be experimentally found out. It expresses the degree of unsaturation of fats. Density of fats. During storage in presence of lipids, fats undergo slow hydrolysis with the liberation of free fatty acids. Short-chain fatty acids have bad smell. Such development of unpleasant taste and odor is known as rancidity, and the fat is said to be rancid. Another cause of rancidity is the reaction between unsaturated fatty acids of fat and atmospheric oxygen, as done by free unsaturated fatty acids. Unsaturated fatty acids of fats combine with oxygen to form peroxides. The peroxides then undergo scission to form aldehydes of objectionable odor. Addition of antioxidants such as vitamin E prevents oxidation by atmospheric oxygen. Acid number. It expresses the amount of potassium hydroxide in mg required to neutralize the free fatty acids present in 1 gram of fat. It indicates the degree of density of fat. Compound lipids. Compound lipids occur in phosphorylated and the non-phosphorylated forms. Phosphorylated lipids or phospholipids are the derivatives of glycerol phosphate and the sphingosine phosphate. Glycerol phosphate derivatives. Glycerol phosphate derivatives are lecithins, which are chemically phosphatidylcholine, cephalins, which are chemically phosphatidylethanol and phosphatidylserine. Other examples are phosphatidylinositol and the phosphatidylinositol phosphate, etc. All these are derivatives of L-alpha phosphatidic acid, in which 2 hydroxyl groups of alpha prime and the beta carbon atoms of glycerol are esterified to 2 fatty acid molecules, while hydroxyl group of alpha carbon atom is esterified to phosphoric acid. Its beta carbon atom is asymmetric and it exists in L form. Therefore, it is known as L-alpha phosphatidic acid. All these are derivatives of L-alpha phosphatidic acid, in which 2 hydroxyl groups of alpha prime and the beta carbon atoms of glycerol are esterified to 2 fatty acid molecules, while hydroxyl group of alpha carbon atom is esterified to phosphoric acid. Its beta carbon atom is asymmetric and it exists in L form. Therefore, it is known as L-alpha phosphatidic acid. In case of plasmalogens, alpha prime carbon atom is interlinked with long chain aliphatic aldehyde. Aliphatic aldehyde changes to its enol form before formation of ether bond. Next, sphingosine phosphate derivatives. They are known as phosphosphingosides or sphingomyelins. 1 molecule of sphingomyelin is composed by 1 molecule for each of sphingosine or dihydrosphingosine, phosphoric acid, choline, and the fatty acid. They are found in nerve tissues. Next, non-phosphorylated lipids. Non-phosphorylated derivatives include glycolipids and sulfolipids. Glycolipids are also known as glycosphingolipids. Cerebrosides, globosides, and gangliosides belong to glycolipids. 1 molecule of cerebroside is composed by 1 molecule for each of sphingosine, fatty acid, glucose, or galactose. Its sphingosine and the fatty acid portion is known as ceramide. Globosides are ceramide oligosaccharides. Their oligosaccharides are made up of glucose and the galactose, and other combinations of hexose and the hexosamine. Granulocytes are derived forms of globocytes with attachment of an acetyl-nuranimic acid. Cerebrocytes and the gangliocytes are found in brain. Next, sulfolipids. Lipids having sulfate groups are known as sulfolipids or sulfatides. Next, derived lipids other than fatty acids. In this section, we will discuss about steroids, terpenoids, and the fat-soluble vitamins, etc. Next, steroids. They are compounds derived from basic structure of 4 fused rings, 3 of cyclohexane and the 1 of cyclopentane. Such fused ring system is known as parhydrocyclopentanophenanthrene. If such structure has one hydroxyl group at carbon atom number 3 and the ASEAN of I to 10 carbon atoms at carbon atom number 17, then it is known as sterol. Examples of sterols are lanosterol, ergosterol, cholesterol, and 7-dehydrocholesterol. Other forms of steroids are bile acids, sex hormones, adrenocorticoids, and the insect hormones, etc. Vitamin D is formed by cleaving ring B of ergosterol upon irradiation by ultraviolet ray. Hence, it is a member of class derived lipids. Next, terpenes. Terpenes are hydrocarbons formed by polymerization of repeating unit of isoprene. An isoprene has 5 carbon atoms and the 8 hydrogen atoms. Some authors use the word terpenoids for terpenes whose methyl groups are eliminated, moved, or containing oxygen. However, organic chemists use both words interchangeably. A monoterpene is composed by 2 isoprene units and it has 10 carbon atoms. Examples are geraniol, limonene, menthol. A sesquiterpene has 3 isoprene units and thus it has 15 carbon atoms. Example is farnesol. Diterpenes, triterpenes, and tetraterpenes are composed of 4, 6, and 8 isoprene units respectively. It has been ascertained that carotenoids are tetraterpenes. Members of carotenoids are more than 600 and they are divided into carotenes and the xanthophylls. Carotenes are hydrocarbons and they are in the subclass of unsaturated hydrocarbons. Examples are alpha-carotene, beta-carotene, and lycopene. Xanthophylls are oxygen-containing carotenoids. Examples are lutein and zeaxanthin. Alpha-carotene, beta-carotene, and the beta-cryptoxanthin serve as precursors of vitamin A for human. Therefore, vitamin A is regarded as lipid under the class derived lipids. On the other hand, vitamin E and the K have isoprene side chain. Therefore, they are also members of the class derived lipids. Next, eicosanoids. Arachidonic acid is the dominant precursor of eicosanoids. It is to be noted that all eicosanoids have 20 carbon atoms. Eicosanoids include prostaglandins, thromboxanes, leukotrienes, and the lipoxins. In our body, prostaglandins serve as leukin hormones. Leukotrienes bring about contraction of smooth muscles. Thromboxanes participate in the formation of blood clot. Lipoxins exert immune regulatory function. Next, lipids complex to other compounds. Lipoproteins, proteolipids, and the lipopolysaccharides are examples. Next, micelle and the lipid bilayer formation. Lipids such as phosphatidylcholine, phosphatidylethanolamine, etc. have charged groups. Such polar groups are hydrophilic, but their hydrocarbon chains are hydrophobic. Therefore, they are amphipathic molecules. Other lipids such as monoglycerides, diglycerides, and sterols have polar hydroxyl groups, and they are also amphipathic molecules. The hydrophobic and the hydrophilic parts of such amphipathic molecules are known as heads and the acyl chain respectively. When cross-sectional area of the head is larger than that of acyl chain, amphipathic molecule forms micelles. When mixed with water, examples are soap molecules. A micelle has few dozens to few thousands of molecules. In a micelle, hydrophobic side chains aggregate inside excluding water, while their hydrophilic heads are in contact with water. On the other hand, when their heads and acyl chain have equal cross-sectional area, they are organized into lipid bilayer when mixed with water. In lipid bilayer, acyl chain of each monolayer occupy inner portion due to their hydrophobic interaction and the van der Waals force, and thus they exclude water. The hydrophilic heads of each monolayer interact with water molecules forming hydrogen bonds. Examples are phosphatidyl choline, phosphatidyl ethanol amine, etc. Next, biological importance. Biological roles of lipids in plants and animals are several, and some important biological roles are discussed below. Fats serve as chief energy reservoir of plants and animals. Fats prevent loss of heat from the body. They also support and protect internal organs by serving as padding substance. Glycolipids, phospholipids, cholesterol, and other steroids are the major structural components of mitochondrial and the other cellular membranes. Lipoproteins are the transport forms of lipids in blood. They are involved in the absorption of fatty acids from intestine. Glycolipids, phospholipids, and steroids play essential roles in the functioning of mitochondria. Cerebrocytes, gangliosides, and the sulfolipids play specific roles in the brain functions. Cholesterol gives rise to bile acids. Cholesterol insulates impulse-generating and transmitting units of brain and the nerve. Wax of animal skins and the furs serve as protective lipids. Plant wax protect leaves and fruits. Ear wax is also an example of protective lipid. Vitamins A, E, D, and K are also members of lipids. These vitamins play a significant role in the vital process. Eicosanoids such as prostaglandins serve as leukin hormones. Functions of other eicosanoids are causing contraction of smooth muscles, formation of blood clot, and the regulation of blood pressure, etc. Now let us come to the conclusion. From the above discussion, it can be concluded that many biomolecules widely differ in structure and function include as member of lipids. The structure of a lipid may be as simple as that of acetic acid, and the complex one is that of a lipoprotein. Fats and oils are neutral lipids, whereas phospholipids are polar lipids. The functions of lipids are diversified. Certain lipids such as prostaglandins serve as leukin hormones. In foods, lipids are present as one of the main components. The essential fatty acids such as linoleic and the linoleic acid are to be regularly obtained from the lipids of foods with their required amounts. Arachidonic acid becomes essential when its precursor linoleic acid is not sufficiently obtained from the diets.